All right. Hi, everybody. I'm going to go ahead and get started with our Chapter 3 lecture. Hopefully, you can follow along with these notes. So chapter 3 is all about the cells. Here you can see kind of just some different cells depicted, and I'll give you a moment. I'll kind of point them out in just a moment, but give you a second to look at those and maybe identify the ones that you see, see if you can identify some of these. Cells comes in all kinds of different shapes and sizes, and it's important to remember that while what we study here is going to be a general cell, and that's what you see right here in the middle is just this generalized cell, that is not the case at all for the actual organisms. There is no such thing as a general cell in any kind of living organism. Every cell is going to be specialized. It's going to have some combination of the organelles. We're going to be studying the structures we're going to be studying today. But some of the few cells, if any, are going to have all of the organelles or an even distribution. A lot of times you'll see cells that have a lot of one particular organelle. Up here in this upper corner, you can see these muscle cells. Muscle cells have a lot of ribosomes and a lot of mitochondria because they're very metabolically active. They're making a lot of proteins. So it's really important for them to have that. Down here in the corner, you can see this white blood cell. This is a type of leukocyte. So it's going to have a lot of enzymes for breaking down different pathogens. This corner, this is a neural cell. This is a neuron. So it's going to be making neurotransmitters, synthesizing those, releasing those, and having to have a lot of membrane permeability with sodium and potassium channels. This upper corner right here, this is a red blood cell. Red blood cells, once they're mature, they get to this size, this shape, they actually do not have any organelles in them. They are completely anucleate. They have no nuclei even. All they have are hemoglobin. They're just filled with a protein called hemoglobin that's used to carry gases around, but otherwise they have no organelles. And these up here are different types of skin cells, just kind of seeing the different shapes that they can take. So again, all of these are kind of more specialized cells. This central one is a generalized cell. And while that's what we study, we have to remember that that's not really the case necessarily in an organism. So this table just likes to summarize cell theory, just kind of jump right into it, because it's important to remember that not that long ago, 150, 200 years ago, people did not understand that there were cells. They didn't understand there were things that we couldn't see that were all around us and make up everything and actually are the functional units of life. The cell is the smallest functional unit of life, meaning that anything smaller than that, while it may have characteristics of life, it may have one or two of them, it not actually is a living thing. It can't metabolize energy, respond to its environment, grow, reproduce, evolve, all those things we talked about in chapter 1. Cells are able to do that. They're this collection of organelles that allow us to process energy and respond and detoxify our blood, all those kind of things. So anytime you hear about an organ and think about an organ's function, you have to remember that it's happening at the cellular level. The action of the kidneys, the actions of the lungs, those are all happening actually at the cell. So that's why we talk about cells are the fundamental unit of life. All cells come from pre-existing cells. They don't just pop out of nowhere. That used to be the belief that things just spontaneous generation came out of nowhere. But now we know, oh no, we can see cells dividing, and we see that once enough cells divide in a contained area, yeah, now it's visible. Now I can actually see it with my naked eye rather than aided with a microscope. Once we started to get better microscopes, we started to change some of our theory. We started getting more scientific about things. We started to see that the cells, no matter what kind of domain they're in, they have some basic compositions. They're all using energy, and they all contain DNA. And that is what's duplicated. That's what's passed down. That's the genetic material for all living life on Earth, living life. So let's talk about some of those common features. All cells share common features, whether it's a bacterial cell or an animal, eukaryotic cell, plant cell, what have you. Regardless of size, they all have that genetic material, some kind of DNA genetic material. Ribosomes, we're going to see ribosomes are essential for making proteins. That's where protein production happens. Cytoplasm and a cell membrane. So you'll see cytoplasm refers to both the liquid and the solid components. So the liquid that everything exists in, which we would identify specifically as the cytosol, plus all the organelles, all the little structures and things. Those together are the cytoplasm. And the cell membrane is the structure surrounding the cell. They all carry out reactions needed to sustain life, things like cellular respiration, protein synthesis, some cells do photosynthesis, right, for producing sugars. And so all of these are essential for life. And cells have this ability to be able to pass things back and forth. And so that's going to be something that we'll see is essential for these chemical reactions. So start with the bacterial cell. There are three main domains of life on Earth based on their cellular characteristics. You have your prokaryotes, your archaea, your prokaryotes with bacteria and archaea, the domain bacteria and archaea, and then you have your domain eukarya. So bacteria and archaea are kind of usually put together, kind of lumped together because they have a lot of similar characteristics. Archaea we don't talk very much about in this class, and even bacteria we don't talk a lot here. You'll get more into bacteria once you get into 112 and beyond. But essentially what you can remember is that bacteria, archaea, they are prokaryotic, which means that they do not have any membrane-bound organelles. There's no real organelles inside of them that are part of an organized system. They're just going to have organelles floating around freely. So you can see some ribosomes in here in the cytosol. You can see the DNA that's that's kind of floating around, but it's not in a nucleus. So there is no nucleus in DNA. There's no nucleus. There's no nucleus in bacteria or prokaryotic cells in general. So that is the defining characteristic for prokaryotic cells, is that they do not have a nucleus, right? Their DNA just floats around in the cytosol. Animal cells are what you're more used to seeing, animal cells, plant cells. These are eukaryotic cells. So eukaryotic means that they have these collection of organelles and specifically a nucleus with the DNA inside of the nucleus. So that is these membrane-bound organelles, meaning that they are contained within this network of systems that's going to allow them to pass structures, pass synthesized molecules back and forth. And again, this large central nucleus that contains and protects the DNA. We're going to go through kind of all of these organelles. We're going to look at their functions, kind of group them and their functions together. But again, recognizing this is a generalized cell, it has all the organelles nice and spaced out. There's nothing on the cell membrane that you can see to make it disruptive. This is not very practical at all. This is kind of like seeing a picture of the Earth with no satellites around it, right? It's just not true anymore. The satellites are surrounding Earth. A normal cell membrane is going to be studded and embedded with a bunch of proteins in it. So we'll see some of that in a little bit. Plant cell, you can see some kind of striking differences. Plant cells have this cell wall on the outside of them, so that's going to help them to maintain their structure and their shape. We'll see that animal cells, we have cholesterol. Animal cells use cholesterol in our cell membranes to help them maintain shape when it gets too hot or too cold. Plant cells have this nice rigid cell wall. So even when your plants, if you're like me and you're bad about watering your plants and they start to wilt, this large central vacuole, right, that's supposed to maintain all this liquid turgid pressure, once this starts to shrivel up, the cell membrane, cell membrane that you can see here, this layer, it's going to start to kind of shrivel in and pull away from the cell wall, but the cell wall will still maintain its shape. So that way, once you rewater your plants or when it rains again, when a drought breaks, whatever it may be, the plant can regain its shape and because that cell wall is still there holding that structure for it. So plant cells, we're going to see that they have the cell wall. They also have that large central vacuole, which we'll see kind of has fluid and enzymes in it. And they have chloroplasts, right? That's another big thing with plants, plant cells, is that they have these chloroplasts for making photosynthesis. Notice that they also have mitochondria. So plant cells, they have to make their own sugar and their own energy. They're doing both of those things together. So let's start talking about the cell membrane. So you can kind of see how the textbook divides up these different organelles and the cell structures. So we're going to start by talking about the cell membrane, and then we look at organelles with protein production, moving proteins around, cell digestion, energy, and then cytoskeleton, some more structural things and communication. So we're going to kind of chunk all of the organelles into these different categories based on their functions. Starting with the cell membrane, here you can see kind of a depiction of the phospholipid bilayer. Let's see, I can zoom in a little bit. There we go. Within this, the phospholipid is the molecule that you're seeing, this round phosphate head and then these tails that are sticking internally. So that's a phospholipid, right? It's a lipid, so that's the class of molecule that it is. So it's a fatty acid tail, kind of like triglycerides have those fatty acid tails, but then it has a phosphate head attached to it. So what's unique about these is that these are called amphipathic molecules. Amphi, like amphibian, meaning they can live on land or water. An amphipathic molecule has both hydrophilic and hydrophobic regions to it. So the heads, the phosphate heads, are hydrophilic, meaning that they are water-loving. Hydrophobic tails, these are the lipid tails. They do not do well with water. They are non-water soluble. These are non-polar, and so they do not interact with water. And so what happens, it's kind of like if you just take, so you're trying to make a salad dressing. You put some oil into some vinegar, some water, and they just don't mix. The oil sits right on top of it. And so you have to shake them up to get them to mix. And then if you leave it sitting out, it'll naturally form this barrier between the two of them. And what happens is that these lipids, these phospholipids, the hydrophilic heads that like water are going to orient themselves towards an aqueous environment. So the heads here, these are pointing in towards maybe the intracellular space. And then these heads on the other side, these are pointing towards the extracellular space. And so you have extracellular aqueous, intracellular aqueous, kind of like you can see in here, right inside of the cell and outside of the cell, intra, extra, intracellular, extracellular. This is allowing the heads, the hydrophilic heads, to point towards water at all times, while the tails are oriented inward towards each other, preventing water from passing through. These molecules will self-organize. They will organize that way. That's why if you just let some vinaigrette sit, it will naturally separate itself to the oil layer on top because of the chemical interactions. These chemical interactions between the hydrophilic, hydrophobic regions of water and these molecules naturally start to repel each other. So this is really helpful because it creates this barrier. And once you have a barrier for something, you can start to isolate, you can localize, and you can really control reactions. You can start to create concentration gradients. We'll get into those, where you have more an uneven distribution of things. And when you start to have unequal distributions, like you can see here, ions and polar molecules, they're repelled, but lipids, small molecules, nonpolar things, they can go through. Once you start to have this uneven distribution, you can start to create things like electrical currents. And so we'll see in the next couple of chapters how that we take advantage of the cell membrane for that. So let's move on. Here's a little bit closer look at this phospholipid. So again, the phospholipid, it has this glycerol chain, this carbon glycerol chain that you see, plus this large phosphate group right up here. This is creating that hydrophilic head, and these two fatty acid tails creating the hydrophobic tails that are going to orient in towards each other with that bilayer. I didn't really mention that. So real quick, you can just kind of see that we call it a bilayer because you have one layer of phospholipids and then it's mirrored with a second layer. And so you have to have a bilayer. If this was just one single layer, then you'd have the heads, yeah, they'd be pointing towards a hydrophilic space, but the tails themselves would be pointing towards the water. And so essentially, if you just have fewer of these molecules, they're just going to make a smaller cell. So your body can change the size of its cells by adding or decreasing the number of phospholipid molecules. As I said, these are amphipathic. There's that word written out for if you want to see it. Amphipathic molecules. You have a non-polar hydrophobic region and a polar hydrophilic region that orients towards the water. Those are good words to learn. Go ahead and memorize those words. Hydrophilic, hydrophobic, amphipathic, polar, non-polar. Connect all those together. So these bilayers, again, their chemical structures, the phospholipids will spontaneously form these bilayers. And let's see if I can get this to open and play for us. I don't think that it will. But I would recommend watching this. You have it on the slideshows. I'll make sure that I put it into the chat as well. This is a really cool video because it shows you how phospholipid molecules naturally will orient themselves just spontaneously into these bilayers when they're put into an aqueous environment. Again, by having that, you now create what's called a selectively permeable layer. And we use the phrase semi-permeable a lot when we're talking about the phospholipid bilayer, the cell membrane, whatever you want to call it, because it's semi-permeable, meaning that it allows some things through, but not everything. Again, lipids, small nonpolar molecules, those things can easily pass through, right? Lipid and nonpolar, because they can diffuse through these hydrophobic tails. But things that have charges, like your ions, polar molecules, things that are going to be typically like large water-soluble proteins, those aren't going to be able to just diffuse into the cell membrane easily. They need to be small, non-polar, lipid-soluble molecules that can easily diffuse in. Exceptions are things like gases, oxygen, carbon dioxide. Those don't have charges, so those can easily diffuse in and out of the cell. That needs to happen. And the cell is going to be always producing channels for things like water, sodium, potassium, chloride, your ions. Your cells, again, make proteins and embed it on the cell surface, so that way they can allow for transport of stuff. So here you can see that. Here you can see these membrane proteins embedded throughout this bilayer to allow for that permeability. So a cell's permeability is directly related to the number of proteins, transport proteins it has on its surface. right? The more transport proteins, the more permeable a cell will be. So some hormones, that's their action, or some drugs, that's their action that we take, prescription drugs that you might take, medications, for a better way to say that probably. Certain medications are going to create more of these transport proteins or downregulate, create fewer of these transport proteins as a way to change cell behavior, change cell functions. So you can just see a couple of these highlighted things like transport proteins that are allowing things to go through, enzymes that are on the cell surface to catalyze certain reactions, recognition proteins. These are a big thing. Recognition proteins, a lot of times it's these ones that have these sugar molecules attached to them, what we call glycolipids or glycoproteins. These are kind of acting like how ships will put their flags and fly their colors and fly their flags to identify themselves from afar, or how on planes they have to have some kind of tail number or wing numbers right on planes to identify themselves from afar. Those are requirements, that way people can keep track of who is around and who's in space. Same thing with cells. Cells want to know, your immune cells specifically want to know what cells are floating around in a space, because if you're something that's going to be causing harm, if you're a foreign cell, a pathogenic cell, a bacteria, something like that, that could be causing harm, then the immune system needs to respond. So your cell, all of your body cells, are going to produce these recognition proteins, these kind of molecules that identify your cells specifically as being native to you, as being, oh yes, these are my native body cells, these are supposed to be here, these are normal cells, don't worry about them. And so the immune system can just kind of keep on floating through. So immune cells will just kind of float through and just really quickly bind these recognition proteins, say, oh, yep, okay, yeah, you're normal, all checks out, all good, keep going. So it's kind of like a way to surveil the body for any kind of intruders, any kind of pathogenic cells. Adhesion and receptor proteins, again, similar things, where adhesion is adhering you to the cells around you or the structures around you, and receptor proteins for things like hormones or signaling ligands that might be trying to manipulate cell behavior, change cell behavior. So the plasma membrane, hopefully you're seeing, is very dynamic. It's allowing for movement. It's preventing things from getting in, and it's interacting with the environment around it. Okay, let's get into organelles for protein production now, protein production. So we're going to get into, once we get into chapters 7, 8, 9, 7 and 8 especially, we really get into a lot of protein synthesis. Is that correct? We'll see. It might be chapter 4. Sometimes my brain gets the classes mixed up and I forget when we learn what stuff. However, we are really going to just do a brief overview of protein synthesis to introduce the organelles that are involved here. So again, I'm not going to ask you right at this point to be learning the steps of protein synthesis. I just want you to start getting familiar with the organelles that are involved with it. So the main kind of overview for protein synthesis is that you start with your DNA, and then from DNA, you make this molecule called mRNA. You can see that we're right in there. So you have a DNA molecule. Oh, oh, come on now. There. Nope, still doesn't like it. There we go. You have a DNA molecule that gets turned into an mRNA molecule. This mRNA is going to then be turned into a protein eventually. Okay, so the DNA is going to be what's contained inside of your nucleus. So the nucleus is that large, it's always going to be the largest organelle in a cell, usually very dark kind of colored. And you can see that it has its own nuclear envelope, its own plasma membrane, same molecule, same molecular structure as the cell membrane. And so this is where the DNA is held, it's protected, right? So it doesn't ever exit the nucleus. DNA will always be contained inside of the nucleus in a eukaryotic cell. You also have this thing called the nucleolus. The nucleolus is this large central circle structure. This is where ribosomes are made. So ribosomes are essential for protein synthesis. They're enzymes that catalyze protein synthesis, and they're made here in the nucleolus and then exit out through these nuclear pores into the cytoplasm. Okay, so starting with our nucleus containing DNA and our nucleolus, we make our mRNA, we make our ribosomes, and then we exit the nucleus through the nuclear pores. Okay. Where then we go to a ribosome. So there are two different kinds of ribosomes. You can see ribosomes here floating in the cytosol, and then you can also see ribosomes here kind of connected and embedded on this structure. These are called the endoplasmic reticulum It's a wonderfully long name. You can see here abbreviated the RER, the rough endoplasmic reticulum. They're called rough because they have the ribosomes embedded on them. It's kind of like they're studded with these ribosomes. So the endoplasmic reticulum, the way that I kind of remember it is, these words kind of these are very long words, and they kind of curl around your mouth. They go to all the different places in your mouth, and that's how these kind of look, right? These are very tubular structures. They're going all around the nucleus, back and forth. Endoplasmic reticulum, right? It's all over the place. And so that's truly how I sometimes remember them, because you have the rough with the ribosomes, and then you have smooth that don't have ribosomes embedded on them. So rough endoplasmic reticulum are for protein synthesis. Specifically, because there are two types of ribosomes, we have these free ribosomes that float around in the cytosol, and then bound ribosomes that are bound to the endoplasmic reticulum. And so they do serve different functions. The free ribosomes are making proteins that are going to be for inside of the cell. They're going to be functioning in the cytosol, intracellular proteins, whereas membrane-bound, your rough endoplasmic reticulum ribosomes, those are going to be going and eventually exiting the cell. They're going to be sent outside of the cell as either maybe a hormone that's being produced or an enzyme that's being produced, something like that. And so they're for extracellular purposes. So free ribosomes, intracellular, bound ribosomes, RER, that is your extracellular proteins. Okay, so now let's talk about how we kind of move proteins around and other structures associated with proteins. So this endomembrane system, it's a connection of organelle structures that allow it to shift. It's kind of like vertical integration. It allows it to take the product from one organelle and shift it to the next, and then eventually outside of the cell. So here, what we're seeing is we're eventually looking at milk production, right? That's the example that we're using from your book, is we're looking at producing milk. Milk, if you know, is a combination of different things. There is some protein in milk, there are lipids, there's a lot of fat, there's some sugar in milk. So it's got a combination of different structures. And so what we start with when mammary glands are producing milk is that they first synthesize the proteins, so DNA to mRNA. That mRNA is going to exit the nucleus into the rough endoplasmic reticulum where the proteins are made. And then from there, it's going to then go onto the smooth endoplasmic reticulum. So here you can see that's what these structures are here. These are the smooth endoplasmic reticulum. And they're going to add a lot of the fats. Smooth endoplasmic reticulum are responsible for synthesizing lipids. They also have enzymes for detoxing certain substances that are digested by the body. And so the smooth endoplasmic reticulum are going to be, in this case, making fats, making lipids to add to the proteins made from the rough endoplasmic reticulum. Those then get packaged into a thing called a vesicle. The cells make things, they make vesicles all the time. A vesicle is like a little Ziploc bag, right? You're trying to move something from one place to another, and you don't want it to get damaged, and maybe it's a little wet, so you want to make sure it doesn't leak onto anything. And so you put it into a little Ziploc bag, and it just transports around really nicely. Cells do that. They use phospholipids, right? So you can see here a phospholipid. They'll take part of their plasma membrane or these structures, the endoplasmic reticulum, the nucleus, these are all made of phospholipids. So that the cell can just pinch off a little piece. That's what you're seeing here. This little bubble right here, these little bubbles right here, these are all transport vesicles that the body uses. Once it makes its protein little lipids, it'll put them into a little vesicle and then float them over to the next organelle, the Golgi apparatus. Okay, so it's like a little stay fresh. It just kind of keeps everything nice and packaged easily so that it goes from one organelle to the next down the line. And then because it's all the same molecule, they can just fuse in with each other. So the vesicles from the smooth endoplasmic reticulum with your proteins and your lipids turn into the Golgi apparatus. The job of the Golgi apparatus is to package everything, finish any kind of final modifications before it exits the cell. So this is kind of the last stop before it exits the cell. I kind of think about the Golgi apparatus like if you, you place your order for Amazon, it kind of you send in all your things that you want, and then at the warehouse, somebody or a robot, whatever it is at this point, is going to collect all the items, and then they put it into a box, they wrap the box, and they maybe add any packaging that might need done for handling and care. They put your label address on it, right, and they put it out for it to be mailed out. That final step of actually putting everything into a box, putting any packaging material, taping it up, putting on the envelope, that's kind of what the Golgi apparatus is doing. It's finishing any final modifications, folding, adjustments that need to be done before the final product, this milk that's being produced, can be exited out through the cell membrane, through, again, a transport vesicle. So this is, again, just kind of highlighting the transport vesicles things. Once they exit the rough ER, they get molded and folded into their final 3D shape. Again, if you remember with proteins, with macromolecules, that final 3D shape is required for function. That all happens in the Golgi apparatus, all of those final interactions and things. I'm sorry, I'm a little behind on my notes. So then the Golgi apparatus, right? You have this processing center. So from the from the smooth ER into the Golgi apparatus, and then from there it fuses with the cell membrane to expel everything out of the cell. Okay, so again, that is just a very overview of protein synthesis, but hopefully you're highlighting these organelles as we're going through. We've so far done the nucleus and the nucleolus, ribosomes, both free ribosomes and ones that are bound to the rough endoplasmic reticulum. We did the smooth endoplasmic reticulum, the Golgi apparatus, and transport vesicles. Chugging through. Okay, so the next one we're looking at are cellular digestion. There are 3 organelles that are involved in eukaryotic cell digestion. The first one are called lysosomes. Lysosomes contain enzymes, ones that are synthesized by the cell, that then you can see them here from the Golgi apparatus, right? Producing these enzymes, modifying them, and then packaging them in the vesicle. These stay here in the cell, though. These lysosomes are going to stay here in the cell, and as cells engulf debris or bring things in, they create this little vesicle that fuses with your lysosomal enzymes and then starts to break any debris down. They break down any debris. I remember lysosomes. These are for general cell digestion. What do you clean your floors with? Maybe Lysol, right? You clean or spray your floors, your countertops, wipe it down with a Lysol wipe. So lysosomes, Lysol, general cleaning for things. Okay, digestive enzymes that are breaking down a lot of large molecules. Go ahead and please note this is not how food is digested. That's through specific enzymes. Lysosomes contain general digestive enzymes. Plants they don't have lysosomes, they have vacuoles, but serve the same function. Vacuoles are also going to have enzymes inside of them that are going to be used for breaking down different cellular debris, different just waste that is produced throughout the body or throughout the organism. Vacuoles also, you can see, regulate the water balance of cells. So again, they can fill up and they can hold a lot of water to maintain what we call that turgid pressure. That's what keeps plants nice and open. When a plant is full of water, its leaves are fully open, and so it can really take in a lot of sunlight and produce a lot of sugar, a lot of glucose in its chloroplasts. When the plant doesn't have a lot of water, its turgid pressure is down, its leaves will start to wilt, and its photosynthetic activity is minimized. So water and the vacuole help to maintain that shape. Something else, the third organelle for digestion are called peroxisomes. Peroxisomes. So these are specifically for oxidizing toxic molecules. So whereas lysosomes and vacuoles have enzymes for just general cell digestion, breaking down cell debris kind of stuff, peroxisomes are going to be going after any toxic substances with peroxide. They're going to be breaking down with peroxide reaction. The way that I remember this, the difference between lysosomes, remember lysosome, Lysol, general cleaning all over your house. Peroxisomes, it has this X in here, this XIO, toxic, X, peroxisomes, toxic, right? The X for toxic. That's how I always remember that. That's the specific reaction that peroxisomes, the specific molecules they're going after are toxins. Okay, the next couple ones are energy-related organelles. This one, again, we're not getting into the actual cellular respiration photosynthetic reactions. That's chapters 5 and 6. I just want you to, again, we're just looking at these organelles. Mitochondria, I'm not going to spend a ton of time with. You're probably very familiar with these. Right, powerhouse of the cell. It's where energy is produced. We'll get into those specifics of the reactions. But you can see its general structure. It has this outer membrane and then an inner kind of very folded membrane to really increase surface area. All of these little folds have enzymes and pumps on them that allow ATP synthase pumps for cellular respiration, ATP synthesis. Mitochondria also have their own DNA. You can see that there is DNA in here. Mitochondrial DNA is passed down through the maternal line. So while all the other DNA in your cells is a combination of both parents, your mitochondrial DNA can be traced back through your maternal line exclusively. It's very interesting. You can also have specific mitochondrial disorders that are passed through the maternal line. The other energy organelle is the chloroplast that you see in plants and other photosynthetic organisms, bacteria, algae. They can all do, some of them can all do photosynthesis as well. So chloroplasts are going to convert sunlight, convert energy into sugar. It's an incredible process. They take this inorganic gaseous carbon dioxide that we're exhaling and then turn that into a physical sugar. It's a really incredible process. And it's actually not the most ancient pathway. We're going to learn that cellular respiration, one of the original cellular respiration glycolysis pathway, that is actually older than photosynthesis. Most people don't quite think that though. So with a chloroplast, where photosynthesis takes place, it is similar in structure to a mitochondria in some ways. You can see that it also has a double membrane. It has 1 and 2 membranes, right? And then it has all these structures in here, and they have a membrane as well. And these membranes are embedded with the enzymes that are necessary for the reaction to take place, just like we have with mitochondria and the ATP synthase enzyme. And so these have very kind of similar evolutionary pathways, and that you have these double layered structures with their own kind of fluid inside. We'll get specifics into the structure of chloroplasts. Some things to go ahead and remember. The space inside of a chloroplast is called the stroma. So the stroma is the fluid, the space kind of all inside of it. And these stacks that you see, these little green stacks, these are called thylakoids. And this is where photosynthesis actually takes place, right? The inner and outer membranes of the chloroplast are kind of for, we'll see concentration gradients, they're for establishing barriers, but it's actually these thylakoids, these stacks here, where the reactions take place. So quick little check-in question. How many of these organelles are in your skin cells? Think for just a second. In your skin cells of a human skin, how many of these organelles do you have? All right, if you said 5, you were correct. Mitochondria, ribosomes, nucleus, lysosomes, and ER, endoplasmic reticulum, both rough and smooth. You'll have all those in your skin. Your skin will not have chloroplasts or vacuoles because we are not plants. Okay, last kind of section of organelles that we have are the cytoskeleton, so I'm going to move through these. A lot of these are for providing structure for the cell, as well as some, collectively, that structure, but also communication and functions in specific ways. So the cytoskeleton, cyto for cell, it's this network of proteins that help to establish the shape of the cell, to maintain the integrity, and to kind of connect different parts of the cell. A lot of times people think that a cell is just this big fluid space where things just kind of float around and may happen to interact, but it's not really true. The cytoskeleton provides a structure for cell movement and organelle transport, so you can actually move things around inside of the cell. There are 3 basic proteins that make up the cytoskeleton structures. You have microfilaments, which are composed of this actin molecule, these actin proteins. This is for contraction. You see this a lot. Intermediate filaments, right? So you can see here these intermediate filaments. They have a varied protein composition, so they're going to have a combination of both your tubular and globulin proteins. And then microtubules are primarily tubulin proteins, these tubulin subunits. And we'll see each of these have their own kind of specific function connected to the type of molecule they have. Microfilaments needed for contraction and changes in the cell shape. So again, those actin molecules are going to help change and adjust cell shape if there is mechanical pressure or distortion. Right now, you're probably sitting on something, or you might be rubbing your fingers together. That's changing the shape of those cells. There are cells that are having to now shift and change their shape and then return back to that shape. And so microfilaments allow for that. Intermediate filaments makes this strong scaffolding for mechanical strength. So again, the fact that cells don't distort out of shape, they don't tear and rip completely when you press on something or run on something or go to hold something or grab something. Your cells have this resistance to that. And so that comes from these intermediate filaments to give mechanical strength. If you fall, if something happens, right, you're not just completely crushed. The last one's these microtubules. Those are the trackways for moving organelles and vesicles. Again, making sure things are going where they're supposed to, kind of like train tracks inside of your cell, organizing that directionality. Two other organelles that are filled with actin molecules, they have a lot of microfilaments, are cilia and flagella. So microtubules, these cilia, these are going to be things that sit outside of the cell. They line your airway a lot of times, and they make sure that nothing kind of sits and collects on top of the cell. So any dust or dirt, anything like that, is going to be kind of swept along the top of the cell surface. So you see these a lot, like I said, in the airway. So people that have like a smoker's cough, people that are smokers, they tend to burn away that airway, the hairs, the cilia, and the airway. And so they have that kind of hacking smoker's cough because their body is always having to get rid of that mucus that the cells are producing. Whereas if you're not really a smoker, your body produces those cilia and they make sure the mucus doesn't stay and collect. They kind of make sure the mucus keeps moving. So if you stop smoking, it usually the cilia grow back after about 6 months or so. And so the smoker's cough can start to wear away if there's not been too much damage. The other one, flagella, these actually help aid in cell movement. So cilia don't move the cell. Cilia move stuff off of the cell. Flagella are going to be moving the cell. Humans only have one cell, one flagellated cell, and that is the sperm cells. Sperm are going to have one tail, one flagellum that allow it to move around and kind of in a whip-like motion. Other organelles, other organisms, they can have cells that have multiple flagella. You can see bacteria that can have dozens of them to help them with their movement. But those are unique to that. Okay, last little bit is cell communication. So cells have to be able to communicate with each other, whether this is to say, hey, there's a virus here, I got an infection. Hey, we need to make more of this protein. Hey, are you hot? It feels a little hot and dry. Should we maybe make some changes? So cells, depending on the cell, it will communicate differently. Plant cells use what's called plasmodesmata. Plasmodesmata are these little channels in between cells, in between the cells of plants. And you'll see the single is called a plasmodesma. So one of them on its own is a plasmodesma. Multiple are plasmodesmata. Right? So that's just that distinction there. You know Latin, right? You got that. This allows for nutrients and chemicals to travel through to adjacent cells, so that way cells can kind of share any nutrition that they might have, but it also makes them susceptible to diseases, because that means that viruses can also travel through. So cells do have a way to send signals back and forth to each other to potentially start closing off those plasmodesma, plasmodesmata, whatever you want to, whichever one you want to use, to try and limit some of that spread of pathogens between different cells. But it is overall more beneficial because, again, it's allowing for sharing of nutrients and biochemicals. If one cell is having trouble synthesizing, or if one cell is overproducing or has an extra full vacuole, it can start to share that. We talked a little bit about signaling molecules on the plasma membrane of the cell surface. These sugar molecules as receptor proteins can help to identify different cells. Crucial for cell communication, absolutely essential for ligand receptor binding. But it's not the only way that cells communicate. Cells also, animal cells, we use what's called gap junctions. So gap junctions are these protein kind of tunnels that are actually allowing ions to pass through. So you can see that in these protein or actual protein transport channels. You see these a lot in the heart. The heart cells need to be able to contract kind of together all at once. And so the gap junctions allow for ions to pass through really fast. So there's no delay in communication with the heart cells contracting, because you want your upper chambers to contract together and the lower chambers to contract together. And so gap junctions allow for that. Tight junctions, these ones right up here, you can see they kind of create this really zip-tied, a watertight seal. So tight junctions, we see them a lot, both in our skin as well as in our GI tract and our abdomen, so that way we're not leaking any abdominal fecal matter or GI content into our abdomen or into the rest of our body. So tight junctions, sheets of cells to form a leak-proof barrier. You can see those nice tight seals. Last one are the anchoring and adhering junctions. Anchoring / adhering, I guess, is a better way. So remember, adhere, adhesions stick to something else. Anchoring and adhering junctions are going to help cells stick to other cells and to the structures that they're around. It's one of the reasons why your heart doesn't just float around your body, your organs don't just kind of disappear and move all around. It's because everything is anchored and adhered to its neighbor, to the stuff around it, to make sure that they're kind of in place, providing that structure and added mechanical strength as well. Okay, if you have any questions, reach out.